In this video, we will cover some of the CAR T cell therapies that are being used on pancreatic cancer and some of the antigens that are under uh, preclinical or clinical trials against this pancreatic cancer. As we know that pancreatic cancer is a solid tumor cancer, so it is difficult for the CAR T cells uh, therapy to act efficiently on the pancreatic cancers against those cancers that are homeopathic, such as this leukemia cancer or the blood cancers. Because in those kind of cancers, the opportunity for the CAR T cells to target the antigen is high compared to the tumor cells, as tumor cells are very condensed. Because of that, it is difficult to target the antigen for which the CAR T cells is designed, and simultaneously the tumor microenvironment will prevent the immune response that are being generated by this CAR T cell therapy. So let us first understand what are the antigens that are being used or that are under clinical or clinical trials. So T cells that are derived from the patients are being used for developing these CAR T cells. So in one of the case, the CD24 T cells is used, and that has produced this around not CD24 T cells. What I mean to say is the T cells with the antigen against CD24 is used, and that is under preclinical trial with 70% efficiencies are being presently observed. And similarly, there are other antigens are being shown here, that is PSCA, CEA, MUC1, mesothelium, FAP, HER2, and among this, we can see that efficiencies are higher for this MUC1 and mesothelium, and both of these are glycoproteins. So this further provides us the pathway or evidence that glycoproteins could be one of the glycoproteins could be uh, one of the most efficient antigen in future, as in the cancer cells there is a change in the glycosylation profile that might leads to generation of some of the glycans that are unique into the tumor cells and that are not found in the healthy cells. And one of the logical reasons could be that the glycosylation might be incomplete in case of these tumor cells, as the cell growth rate is higher. So uh, glycans might come on the cell surface, or glycoproteins might come on the cell surface without complete or proper glycosylation. That might lead to improper glycosylation that has been observed on MUC1 and this mesothelium. In the MUC1, the antigen that are produced in the case of cancer cells is TN antigen and STN antigen, and some studies are also been carried out on those kind of antigens on a mice model, and that has shown that has shown a positive result, and simultaneously no cytotoxicity has been observed in that studies. And the other approaches that have been focusing on in the case of this pancreatic cancer to target the pancreatic cancer or tumor cells is to engineer the CAR T cells in such a way that intracellular part of this CAR is chimeric antigen receptor will be we can say optimized in such a way that it will produce a more stronger immune response whenever the antigen is detected by these T cells. And some of the signaling domains that are being worked upon is ICD, or we can say intracellular signaling domains that are being worked upon are ICOS and 41BB. That will produce the cytokines that can help to kill the tumor cells that are being recognized by these CAR T cells based on the antigen against which these T cells are designed. And the other approach is that the T cells can be designed so that it can detect more than one antigen, as the example that is shown here, that these CAR T cells can detect three antigens. One is IL-4, PSCA, TGF, beta. So this can target tumor cells. This can target different tumor cells that has different kinds of antigen present on their cell surface. And the next one is the case of using different T cell population uh, based on the individual performance. And different T populations can be used. Different T population can be used to target different kind of tumors at different parts of the body, or different kinds of tumors that are present within the body. So, for example, the case that is shown here is that these tumor cells, this CAR T cells can target this CAR T cells can target this CD26 into a specific region where the CD26 expressed tumor cells are more prominent. So we can decide the T cells based on the location of the cancers that are being identified, so that the efficiency of the CAR T cells can be more efficient on that place. So these are some of the some of the ideas or approaches that are being going on to treat pancreatic cancer or solid tumors using CAR T cell therapy. If you need to understand more about these studies, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.